Greetings, one and all present here. I am Anagha Pradeep Kumar, a first year graduate student under the supervision of Dr. Lord Abbey at Dalhousie University. And I'm here to present on the topic Kale plant response to marine-derived biostimulants. Let me tell you, plant nutrition is vital to deliver correct plant nutrition at each plant stage to maximize plant production. And since 1970s or the Green Revolution, a lot of chemical fertilizers have been added to the soil as soil supplements. This, along with various other anthropogenic activities, have caused threat to agriculture industry, which ultimately impacts the food and nutrition security. So now we are looking at reversing the damage that we have caused. For this, we have to adopt regenerative and sustainable agriculture practices. As we continue to explore new ways to improve sustainable agriculture practices, we are finding that sometimes the most effective solutions are found at the most unexpected places. So in this presentation, I'll be discussing on how sea minerals and fish hydrolysate, both being biostimulants, can offer a promising alternative for enhancing kale seed germination and growth. I talked about biostimulants. Let me tell you what biostimulants are. Biostimulants are substances of organic, inorganic, or microorganism in origin, which, when added to the plants in small quantities, can help plant decrease the stress, can increase the growth and productivity of the plants. Now, let me tell you why ocean can be considered as a resource for biostimulants. Almost 91 million tons of fish is harvested every year, and out of which just 30% of it is transformed to fish meal, and possibly 50% or even more is thrown back into the ocean as fish tissue processing waste. These waste contain significant amount of bioactive peptides and amino acid which can increase plant growth and help in human and animal feed supplementation benefits. So what is the way that we can use this fish waste instead of dumping them? Can we recycle them by composting? We can, but there is low protein recovery. So to facilitate the recovery of essential nutrients, we are looking at value addition of products, and there comes fish hydrolysate, which is a value added product of the waste. When I talk about biostimulant market, humic and fulvic acid constitute half of the biostimulant market, which is a major limitation. People don't know what biostimulant options they have. And then the effect of sea minerals is unexplored. And there have been very few studies on corn, winter wheat, and lettuce and other crops. So we need to explore it to other crops and the studies. And another limitation of the biostimulant is that they do not have a consistent global definition. This causes a lack of proper regulatory framework, which results in the manufacture and sale of questionable products in the name of biostimulants. This results in the adoption rate of biostimulants in the global market, and it is going down. But even though there are a lot of limitations, the global market of biostimulants is actually growing at a compound annual growth rate of 10% and is expected to reach 4.1 billion US dollars by 2025. So there comes my objective of determining the effect of sea minerals and fish hydrolysate on germination, seedling, and plant growth parameters. For this, I had my materials and methods are here as follows. I followed a completely randomized design. I had 4 treatments, which are liquid biostimulants, out of which 3 were sea minerals and 1 was fish hydrolysate. The sea minerals, I have named them as SM1, SM2, and SM3, and fish hydrolysate as FH. I had 5 concentrations of 0.25, 0.5, 1 and 2%, and these were applied on the plants. I had 4 applications, and my treatment plant was Kale and the variety, the dwarf green curl. I did my statistical analysis using Minitab, and I used one-way ANOVA and general linear model, and means were compared using Fisher's protected LSD. Coming to my experiment, I had 2 types of experiment. One was seed germination pouch assay and one was greenhouse trials. Let me take you to my seed germination pouch assay. These are the results of germination pouch assay. As you can see, my control versus all my treatments are placed side by side. Here in the picture, if you notice that the control plants are growing at the roots and shoots are in a good level. But when you compare it with other treatments, you can see that the sea minerals and the fish hydrolysate produce plants with better and more number of lateral root growth, which results in better nutrient absorption from the soil and results in overall shoot growth and development of plant. To make the picture clear, I have them side by side. As you can see here, control versus sea minerals 1, sea minerals 2, sea minerals 3, and fish hydrolysate. When you look at all these pictures, you can see that the roots are better and the plants are definitely doing better. The statistical analysis results showed us that sea minerals 1 and 2, which you can see as a black bar graph of 0.25, did exceptionally good and increased root and shoot volume when treatments were applied. When you look at shoot and root length, again you can see that sea minerals 2 and fish hydrolysate was effective in increasing the root and shoot length. When you look at the root and shoot surface area, again, sea minerals 2 at 0.25% and fish hydrolysate both at lower concentration was found to be effective. Also, when we were analyzing germination rate, the sea minerals 3 were found to germinate faster and better compared to the control. Now, I'll take you to my greenhouse trials. The results of the greenhouse trials, as you can see here, is control versus all my treatments. And you can see it is very clear that the fish hydrolysate was doing much better compared to the control. The plants have more more number of leaves, better and greener leaves compared to the control. When you look at sea minerals 1, 2, and 3, they are also doing better. But among all treatments that were applied to the plants, fish hydrolysate did outperform the other treatments. And these are the statistical analysis results of the greenhouse trials. As you can see here, fish hydrolysate at higher concentration, which is 2%, had the total chlorophyll per unit area the highest. And when you look at the plant height and the number of leaves, again, fish hydrolysate was doing much better compared to the rest of the treatments. This is the total number of green leaves. Again, total number of green leaves was also found to be better and more for fish hydrolysate. This concludes both my experiments, and my conclusion and impact would be application of 2% of fish hydrolysate significantly was able to increase the plant height, number of leaves, and chlorophyll content of kale plants. And when you look at the germination pouch assay, 0.25%, which is the lowest concentration of sea minerals 2 and 3, was able to increase root and shoot length, surface area, and volume for kale seedlings. Hence, in conclusion, my research on kale seed germination and seedling growth responses underscores the importance of sustainable agriculture and the need of urgent action by humans to adopt more environment-friendly conscious farming practices. Whether we humans live near the coast or far inland, we are the part of the problem and we are the solution to the problem. As we look towards a sustainable future, it is critical that we continue to explore innovative agriculture practices and solutions that prioritize the health of our planet and well-being of future generations. Together, we can create a more resilient and sustainable food system for all. Thank you so much.